Welcome back. Let's go through a brief overview of the synapses. So the axodendritic site is going to be the most traditional synapse. So the axon is going to make a connection with a dendrite, and it's the most common type in the CNS. The axosomatic is pretty rare because the axon will contact the soma or the cell body directly. Axonic is even rarer because it's very, very rare. It's usually only really due to injury where there's some proliferation or regeneration, reneuralization that's needed. But the axon needs to synapse with either like the cell body or the dendrites in order to release its vesicles. It's not really able to do that with the axons because the axons are usually surrounded by myelin. Dendritic are even rarer, and those are only in the retina and the olfactory bulb. So here is the way the synapses really form. It's not just like one little connection, there's a bunch of connections. In the neuromuscular junction, it's quite different because it infiltrates the skeletal muscle fibers. And in the neuroglandular synapse, this is more kind of what people think neurons and axons would be, right? Or axons and dendrites or synapses would be. So one per like gland cell. Okay, so the chemical synapses use neurotransmitters, and it's a one-way conduction. It's slow. Electrical synapses are much faster because they just kind of like work via the concentration gradient. It has a bipolar conduction, so it can go either way. And it's predominant in the smooth cardiac muscle, rare in the CNS. So in the chemical synapse, there is either like a membrane potential alteration, biochemical cascade due to the G proteins, or it can actually change gene expression itself. Okay, so we already went through like the clear core and dense core, but the dendrites will end in these terminal boutons or synaptic knobs, and those will contain the synaptic vesicles, the mitochondria, and the lysozymes. The mitochondria in order to reuptake, remember? So clear small vesicles will make the small peptides, so acetylcholine, GABA, glycine, glutamate. The small dense core vesicles will be responsible for making catecholamines, and the large dense core vesicles will make the neuropeptides. Neuropeptides are able to, travel further. Okay, so dense tufts are filamentous projections, and these are in close contact with the vesicles in the active zone, and it plays a role in the exocytosis. Active zone contains the calcium channels, and that's where the neurotransmitters are released. Any problems with the calcium channels in the active zone, it would be a result of like the Lambton-Eaton syndrome. I think I'm saying that right. Okay, so the synaptic cleft isn't actually empty. It's filled with ECF fluid and polysaccharides. The neurotransmitters are going to be released, and they diffuse and dock onto the postsynaptic receptors. Neurexins are proteins that are attached to the presynaptic membranes, and it binds to neuroligands, which are on the postsynaptic membrane, and they basically just form the bridge to hold the synapses together. And they also work as a ligand for the beta neurexins. And the postsynaptic vesicle will contain the dense areas, and that's specialized for the synaptic transmission. And the receptors are going to be clustered in the postsynaptic densities. Okay, so the synaptic potential have longer duration than action potentials. The excitatory postsynaptic potential occurs when the depolarization of the ligand-gated channels in the postsynaptic cell, and that's made by acetylcholine or norepinephrine. It's going to cause the influx of sodium in order to depolarize the axon, or calcium in order to release acetylcholine, and the closure of the potassium channels. The inhibitory postsynaptic potential works by hyperpolarization, and it's done through GABA or glycine, and it's caused by an influx of chlorine and potassium, and the closure of the sodium and the calcium channels. Electrical synapse go from pre to post only, and there's a delay because the neurotransmitters need time to be released and diffuse across the membrane, or not the membrane, sorry, the cleft, and bind to the receptor. So fatigability is dependent on the amount of neurotransmitter available. Remember, in myasthenia gravis, with each action, with each like somatic movement, they use up their acetylcholine, and there is going to be like less and less and less available every time they move. So there is a couple ways that electrical synapses work together: by convergence or divergence. So like convergence, a bunch of synapses work together, converge into one, and that makes the signal stronger. Divergence, it's going to relay the one signal to a bunch of different like cells, so kind of in the muscle. Summation can be either spatial or temporal, so a lot of signals coming from different axons will come together, merge, and form one big action or one big signal. Temporal is going to be many signals kind of working all together onto one. And they can either be excitatory or inhibitory. Okay, so synaptic blocking agents are going to work best with long chains of neurons with multiple synapses, because you can block one chain and everything downstream of that one neuron is going to be blocked. So hexamethonium tetraethylammonium salts, they resemble acetylcholine, and they inhibit their transmission across the synapse. Nicotine will resemble acetylcholine, and those are not removed by acetylcholinesterase. Instead, there's going to be a prolonged depolarization and insensitivity to further stimulation by normal acetylcholine. Procaine, it's one of the anesthetic drug, and it inhibits the release of acetylcholine from the preganglionic fibers. That's it for now. Thank you for your attention, and good luck studying.